So it's 10 o'clock. As a Swiss company, we want to be precise. So I will welcome everybody to this new webinar of the Neurolite Academy. I am Florent. I'm the CEO of Neurolite. I will just introduce briefly our company, and then we will switch to the actual presentation. So Neurolite is a small team, about 15 people that have a common passion, the field of neurodiagnostics, neuromodulation, and neuroscience. So our products are really around these three pillars. In neurodiagnostics, more precisely, we deal with systems from several manufacturers worldwide and deliver them to our customers in hospitals and private practices and clinics in the country, in Switzerland. These are ENG systems, evoked potentials, EEG, sleep studies, and transcranial Doppler. In neuromodulation, we propose transcranial magnetic stimulation, transcranial electrical stimulation, be it TDCS or TACS or TRNS, and even a specific device to fight migraine. And then in the field of neuroscience, we offer a variety of solutions, systems around NIRS technology, which we will talk more in details today, but also EEG and eye tracking, among others. And of course, for all this, you need accessories, electrodes, head caps, gels, paste, everything that we also propose, of course. We also provide the service and the support locally. We have a team of 4 technicians based near Bern in our headquarters. And we support all our customers, install base, which is about 500 systems right now in the country. And yes, we do the repairs, we do the service, we do the installations, of course, ourselves. And we also accompany our customers with a full offer of trainings. So at the installation, of course, explaining how devices work, but also more with webinars like today to spread the word about several techniques, to make sure that more people are aware about the possibilities of these several techniques. Today, I will, after, give the word to our NIRS specialist, Coralie Debrac, who is also working at the University of Geneva, where she has achieved a PhD, especially with NIRS research. She's also part of the Neurolite team, specifically to support our research customers that work in the field of NIRS, but also in other modalities like EEG, TMS, and TDCS. So I give you the word, Coralie, and wish everybody a pleasant webinar. Thank you.
So good morning, everyone. So as just said by Florent, I'm Coralie, and today I will discuss with you about the use of Near Infrared Spectroscopy, NIRS, in naturalistic environment. If you have any question during the presentation, write them in the chat, and I will respond to this question during the Q&A session. Let's start the presentation with a quick overview of the principle of NIRS. NIRS is based on an old principle named edge transillumination, described for the first time by Richard Bright in 1831. The transillumination is the passage of light through the body. In other words, NIRS is similar to a torch lighting up blood vessels from brain or muscles, allowing us to assess the concentration of oxygen, which is contained in the hemoglobin. Despite its pretty old principle, NIRS was used for the first time in research in the 90s, making NIRS the youngest neuroscience technology to assess oxyhemoglobin. For this matter, NIRS do not use any light, but near infrared light between 700 nanometers and 950 nanometers, based on the work of Franz Jobsis in 1977, considered as the father of NIRS. This near infrared light is emitted by an optode for optical fiber and received by another one, constituting one channel. As for the measurement of a black hole in space, based on the difference of the near-infrared light emitted and then received, because oxygen will be absorbed by the near-infrared light in the middle of the light path, namely the banana shape, NIRS can assess using a Beer-Lambert-Low transformation, transforming the measurement of light absorption to oxygen concentration, the level of oxy and deoxyhemoglobin for each channel. Usually, in the literature, distance between one emitter and one receiver is around 3 to 4 centimeters in adult and around 2 centimeters in young infants. Moving forward now to our topic of interest, which is naturalistic environment. But why it is so important to perform experiments in such conditions? Because for some experimental tasks, measure brain or muscle oxygenation as if the participant was in the real life is the best way to investigate the processes at play in this task. It is especially important given the necessity of replicability in previous research in psychology or neuroscience domains, for instance. And to illustrate my word, I can give you an example. One friend of mine wanted to investigate the processing of synchrony between two musicians when they were playing violins together. For this matter, one musician watched video of violinists using a screen in fMRI scanner. My friend obtained really cool data, actually, that will be soon published, hopefully for him. But we can question how this data is really reliable to the processing happening in the real life when the two violinists are actually playing together during a public concert. I strongly believe that is not exactly the same story. And to illustrate my thought, I will play now a short video on NIRS using hyperscanning condition with two violinists playing together. I leave you with a beautiful music background also. So here we go. So following this, perform experiment in naturalistic environment can be also a matter of ethics. Think about animal research or task in children with autism, for instance, how it is ethically correct to constrain a non-human animal for research purpose, or how it is ethically correct to ask a child with an autistic syndrome to stay calm in a fMRI scanner when you could have the possibility to directly investigate your research question at his or her home, or even at school, for instance. Here are the main reasons of the increasing number of publications using now a more naturalistic approach. But to do so, a device needs to follow some criteria. First, the device, or at least the device cap, must be wearable, then easy to wear for the participant, but also easy to adjust for the experimenter. So no long preparation with white gel and electrode as for classic electroencephalography, EEG, for example. Second, for most cases, the device might be wireless in order to allow free movement to the participant. Following this, and because participants will freely move, the device must be poorly sensitive to motion artifacts in order to reduce confounding factors to the measurement. The device must be non-invasive to follow the new ethical rules, obviously. And finally, in order to use comparative approaches, the device must be mainly suitable for adults and children, but also suitable for animal research. Hopefully for us, some NIRS systems include all these criteria and can also be combined with other wireless devices such as EEG or TDCS, transcranial direct current stimulation, for instance. The first system is a BRIGHT, enabling online and offline measurement of brain oxygenation with 44 channels in adult, but also in infant. And with this specific device, your participant could make some exercise at home, for instance, and you could take back the data the day after directly from the device with an USB key. I will now play a short video showing you how the BRIGHT system is perfectly suitable for experiment in young infants in naturalistic conditions. So here is a little boy playing with his dad. And you see now the NIRS cap in open and perfectly adjust for the child health. In this example, the experimenter chose to cover all the left hemisphere with the optode, but as you can see on the cap, you can place the optode everywhere on the scalp. You can even cover the full head if you are interested by the whole brain mechanism. So in blue and red, the oxy and deoxyhemoglobin signal in the NIRS software named OxySoft. And you can see that the child doesn't care at all about the NIRS, which is really important to have natural behavior. The child is just focused to play with his new car. The second system is Octamon, enabling online measurement of brain activity, also in adults and children, but using 8 channels this time. This system is really light and originally designed to measure prefrontal and frontal cortices activation. But now there is new cap enabling to the 8 channel to be placed everywhere on the scalp. Again, to illustrate my word regarding frontal measurement with the Octamon, I will show you a short video of mine, actually. A few years ago, with my lab at the University of Geneva, I make an intervention in a private school. For this purpose, I use the NIRS Octamon all the day on different children listening music. And I'm showing you this video because it's a good illustration, I think, of how it is easy to set up the Octamon device. So I play the Octamon and then a few seconds on the child health, and then explain the oxy and deoxy signal to them and their parents. And the little yellow suitcase in the background to transport the NIRS system. So really, really easy to set up and to transport everywhere. Finally, the Portalite and Portamon system, both using 3 channels to measure brain, but also muscle oxygenation, TCI, tissue saturation index, in adult, infant, and interestingly, non-human animal, we will see later in the next slide. Both devices are extremely light and adaptable. In order to close this part on the different existing NIRS system, I will play again a video illustrating the use of Octamon, Portalite, and Portamon in outdoor conditions. So here's the Octamon. In order to assess oxygenation of leg muscle in a running subject, for instance. Or still the Portamon for muscle oxygenation of leg, but now in biking condition. So you see the Bluetooth and the dongle key for the software. And now you can biking at more than 100 meters from your experimenter. Here an illustration of the Portalite system to measure brain oxygenation with a lithium battery attached to a belt. And you can stick easily and directly the Portalite system to the front head. Now the experimenter is synchronizing both devices with the remote. You see the oxygenation, oxy and deoxy signal. Here is an illustration of both Portamon devices for the front head with the end band for hyperscanning measurement, for instance. Here we go. Let's now moving forward with the application of this NIRS system in research. Starting with what I love to call extreme research, namely research in wild animal in water. For this purpose, the author used as participants 4 seals for which, after habituation phases, of course, they place a Portalite system to measure oxyhemoglobin and TCI from the right parietal cortex of animal during dive. Here are the results. So the green light represents the blood volume, the black line represents hemoglobin oxygenation, and the red line represents TCI. The author demonstrates the suitability of the Portalite to assess the decrease of oxygen during the descent and the increase of oxygenation when seals were surfing at the surface, but also the modulation of oxygenation during the dive. And to follow the application of NIRS in extreme environment, I will show you again a short video applying NIRS to assess frontal activity in aircraft pilot. This video was made in an aircraft simulator, but I know that some labs in the south of France are actually using NIRS during real flight.
Superior 32, speed 256, heading 138, altitude 12,400, vertical speed plus 1500, over.
A second interesting application is the use of NIRS in children. Here, to investigate the difference of muscle oxygenation during a walking test in full-term and preterm children. For this purpose, the author placed the NIRS potamon on the right medial gastrocnemius of preterm, so the leg muscle, and full-term infant aged from 3 to 6 years old. Then the experimenter asked to the children to walk as fast as possible during 6 minutes. Here, as a result, with a 6-minute walk on abscess, the concentration of deoxyhemoglobin on the ordinate, the black square represents the preterm infant and the white circle, the full-term one. And you see that between 3-minute to 6-minute walk, preterm infants have significantly more variation of deoxyhemoglobin in their right leg muscle comparing to full-term infants. This result suggests a greater change in peripheral muscular oxygen extraction associated in this paper with slower walking speed and stride length. The author concludes to a muscular maladjustment and reduced functional capacities of children born preterm. At the opposite of this paper in infants, NIRS can be used in elderly people research, such as this recent study on the involvement of prefrontal cortex in stair climbing conditions. For this purpose, the author asked 20 participants around 72 years old to 1. Respond to a question while ascending or descending stairs, or 2. Climb stairs without saying anything. The involvement of the prefrontal cortex in this simple and dual task was measured using the NIRS octamon. In this figure, SM represents a condition in which the participant was only climbing stairs for simple task, and DT represents the dual task condition. The author revealed an increase of oxyhemoglobin concentration for the dual task comparing to the simple task. And for that, the author suggests that executive processes are less efficient during dual task and then need more effort, oxygenation, to the prefrontal cortex. NIRS can also be used in daily life situations, such as back oxygenation during long-term driving. For this matter, the author used the NIRS spotlight to assess the oxygen concentration changes in the erector spinae muscle in the back when the participants were driving a vehicle on a highway for a long time in 2 different driving postures, A in blue or B in green. So here in blue the posture B and in red the posture A. In abscess, you have the time, and in ordinate, the level of back muscle oxygenation. And the author demonstrated an increase of oxygenation in participant back, when driving with the posture B, suggesting a lumbar discomfort in this specific position. Finally, if outdoor conditions are so extreme or even impossible because of your population, for instance, you could also use virtual reality to approach naturalistic conditions. Here is an example with elderly people with Parkinson's disease. For this purpose, the author used NIRS, a fixed device called Oxymon, and transmagnetic stimulation, TMS, to assess the effect of TDCS on dorsolateral prefrontal and premotor cortices in Parkinson participants while doing physical exercise with video game. The study is still in progress, but already promising. Finally, to wrap up this webinar, on NIRS in naturalistic environment, we saw that NIRS is a really good candidate for performing experimental tasks in naturalistic environment, from extreme outdoor condition to virtual reality. This can be explained by the fact that NIRS is a non-invasive technology, perfectly wearable, wireless, and poorly sensitive to motion artifacts. NIRS has also several advantages for such research, such as the measurement of brain and muscle oxygenation, the possibility to combine other portative devices, EEG, TDCS, electromyography, etc. And also really interesting, NIRS is suitable for human research, of course, adult and infant, but also for animal research, such as seal or non-human primates. On that, I would like to thank you and to mention the next webinar of the Neurolite Academy happening the 20th of May at 6:30 on the use of non-invasive brain stimulation in psychiatry. If after our Q&A session you still have some question or have interest for one of our devices, NIRS of course, but also TDCS, EEG or TMS, you can always reach me by email or by phone. You will also receive soon an email from Neurolite to have your feedback on the today presentation. And now it's the time for your questions, so write them in the chat if you have some. I'm really happy to take every question that you have. So I don't see any question for the moment. Don't be shy. So I'm waiting 1 or 2 more minutes in case suddenly you have a question. But in case, again, you can send me an email if you want really precise info or publication. Oh, thank you. How many subjects can be monitored simultaneously by the Artinis system? So usually you can, in hyperscanning, do 2 subjects. That is the base of hyperscanning. But I don't see any limit, at least. You can do whatever you want. If you use, for instance, MATLAB or Python to use external trigger. I know in the Netherlands, they did with Artinis devices during a public concert with almost all the musicians with NIRS devices. So sky is the limit. How many devices you have, you can do your hyperscanning system. But the base is 2. It's the easiest one, obviously. Thank you for your question. Does someone have another question? So the device can get the data in real life and use it right away, or does it need pre-processing of the data? So, so thank you first for your question. So as for EEG, you have 2 ways to do that. You can use with the Oxysoft software, so the software of the NIRS of Artinis, pre-processing data in live, or you can just upload the raw data and apply a posteriori a pre-processing. So it's as you wish. But if you want to use right away the data, as you suggest, yeah, you can apply in real time a pre-processing. So every like heartbeat artifact, for instance, and movement artifact also. Thanks. Is there some delay, if any? Yeah, it's kind of really millisecond. So for that, I don't know exactly the delay, but it's extremely short. It's around, it was what? Yeah, 50 milliseconds, something like that. But you can easily saw it. For instance, with an LSL port, for instance, you see when you send a trigger, for instance, and when we receive it, but it's extremely short. Thanks. Do you have more questions? So I'm waiting a little bit. What is the working distance? As shown in the outdoor cycling sport, it seems the device works for very long distance. The data are transported to the device online or upload after the cycling. So depending the device. So for the bright devices, so the huge one, or you can do 54 channels, there is no matter of distance because the data are directly go in the little kind of battery or SIM card inside the cap. So, and after you can take the data back 2-3 days after if you wish. You don't need to be close with your computer. And with other wireless devices, so Portalite, Portamon, and Octamon, the experimenter, at least the computer of the experimenter, have to be around 100 meters to the participant. But 100 meters is still give you still a huge distance. But that is the maximum for these devices. So if you want to be free, completely free, yeah, the best option is the Bright system. Thanks. More question? How sensitive is the overall system? Could one, for example, wear a hat or helmet or a head-mount display virtual reality system? Yeah, actually, there is some study like that. They start to use the NIRS in virtual reality with the VR. It was a HTC, I believe. Yeah, the HTC one. So it's perfectly compatible. Yeah. Especially that there is some VR devices now working with NIRS, so they try to combine together. But it's okay. And again, the motion artifacts are pretty obvious in NIRS. So NIRS is quite less sensitive to motion artifacts, so even head cap or, I don't know, head movement or arm movement like me. But it's definitely not the same frequencies as the signal that you want to target, typically oxyhemoglobin. So with a bandpass filter, it's with Homer2 or Homer2, sorry, or NIRS SPM, the two usual toolbox for the pre-processing. It's really, really easy to remove this artifact in case you want to use it in more extreme condition, maybe in virtual reality, depending on your experiment. Do you have more questions? Last chance, for now, at least. Okay, so if I answer all your questions, I will close this webinar today. Again, you can always send me an email, and I'm really happy to answer or give you access to the relevant literature if you need it for your experiment. Or I can come to the lab also to help you with this matter or to discuss with you about what you want to do. And I would like to thank you for all your questions to attend to this webinar also. And on that, I would like to wish you a really good day, hope more sunny than in Bern. And thank you very much again for your attendance. See you next time. Thanks.